Yeah, it's a really exciting time, in Alzheimer's research and here at AIC specifically. At Biogen, we've been committed to this space for a long time, and particularly thinking about the two different proteins that we believe drive disease, the aggregation of amyloid beta, but also, as you mentioned, the aggregation of tau. And so we have a big focus here at this meeting talking about tau in a couple different ways. First of all, with our BIIB program, where we showed the baseline characteristics in our CELIA study, as well as work that we're doing on innovative biomarkers to help drive the field forward with tau. So all of that, I think, will help inform folks about, first of all, the exciting things happening in amyloid, but now the next wave of tau therapy. Targeting tau is something that we've known for a long time, that this aggregation of this abnormal protein is coincident, with the clinical decline for patients. But getting a drug at that target has been pretty tricky because it resides predominantly intracellularly. And so there are a few ways to go after it. One way that we're pursuing with BIIB080 is to just knock down production. And so BIIB080 is an investigational but first-in-class antisense oligonucleotide that knocks down all isoforms of tau, and therefore will bring down levels intracellularly. We know in our 1B study, when we administered that to patients, we saw knockdown in the cerebrospinal fluid and then an impact on their tau PET imaging. So we saw manipulation of the biology there, and it was a small sample size, but we also saw trends to improvements in memory scores. So that led us to have the confidence to basically go into our larger study, the CELIA study, where we put the baseline characteristics out here at the Congress. And that's a much larger study looking at this intrathecal administration of our antisense oligonucleotide. And we're administering it either twice a year or 4 times a year to see if, when we administer it, knocking down all those forms of tau actually correlates in a larger patient population with impact on imaging biomarkers, on tau PET, but most importantly on clinical efficacy. And so we completed enrollment of that study last year, and we're hoping to read it out because it's an 18-month clinical endpoint in 2026. So that's a big way in which we're doing that. There are other approaches to tau knockdown, you'll see, but to get it intracellularly, you'd either have to go after a small molecule based approach, which can, basically get intracellular, intraneuronal, and affect aggregation, or potentially even block propagation. And there's some folks working on vaccines or passive immunization approaches. But our take on it is to go for the antisense route. We're fortunate at Biogen to have been, on a long journey with the amyloid therapies and learned a tremendous amount, and hopefully we can, take that knowledge and bring it to bear into the tau space. It took time for us to figure out that relationship between figuring out which patients to bring into clinical trials, and our work in amyloid PET allowed us to figure out the right patients to put in, then have the right drug, then hit the target hard enough, bringing down the centiloid thresholds to then figure out what the efficacy looked like. And so that arc, that sort of chain of translatability, is something that now we're figuring out with tau. And BIIB080 hopefully be one of the first assets that allows people to figure that out, because we know we're hitting the mechanism in the CSF. We know from the 1B, and we'll show hopefully again in the CELIA study what we're doing on the PET. And then we need that clinical correlation. So it's still earlier days to do that. We haven't established that, but our work, I think, particularly in the biomarker space, we have a very high fidelity tau tracer that we have on our PET scans, which will allow us to study that. We also made a specific intentional decision to study in our BIIB080 CELIA study all forms, basically. We're not making we don't have a preconceived notion of how much tau burden you have to have in order to see if you can benefit. We're gonna learn, in this trial. And so that's a very sort of CSF and then PET-based imaging. Of course, we're excited eventually as the amyloid field's moving to blood-based biomarkers. Hopefully, too, you'll see tau moving in that space, too, for fluid biomarkers. It could be the things that we're seeing that are intriguing to us are the CSF, MTBR, and we have some data that we'll present at this Congress looking at that, as well as some other peripheral tau biomarkers. We just still need that correlation with an interventional study to show us what it does to the clinical endpoints. But it's coming, and I think all our lived experience of doing it with amyloid, we can bring to bear to do it with tau, and hopefully in a more expedited and informed way.